Hello everyone, I'm Shin from Korea. We recently discovered a new species candidate from hypersaline pond in Korea. These new species populations belong to the genus Schmidingerothrix, which is morphologically different from the congenus and phylogenetically positioned as a sister group relationship with the close congenus. Schmidingerothrichidae salina and Schmidingerothric salinarum, based on the 18S ribosomal RNA gene sequence analysis. I would like to introduce to my student Novia and Ratih, who participated in this study. Novia did the morphological analysis and Ratih did molecular analysis. They will explain more in details about their result and answer your questions.
Hello everyone, I'm Cahyani Novia. I would like to explain more in detail the morphological aspect of our new candidate species of Schmidingerothrix.
Hello everyone, my name is Ratih. I would like to present about the phylogenetic aspect in the new candidate of new species from genus Schmidingerothrix.
So our new candidate species of Schmidingerothrix we found in the Kusuma pond water in Korea. The morphology of live specimen were studied by the bright field NDIC microscopy. We examined the ciliate by the protagol impregnation. So here our species study. This species have a size in vivo 64 until 101 times 10 until 30 micrometer. This cell have oblong to sigmoidal shape and has short distinct tail. The cell contractile, highly flexible and colorless. The cell lacking of cortical granule and dorsal bristle. This cell have two prominent frontal cirri and two rows of frontoventral cirri. And in the adural zone membrane, we can found here the buccal lips. Interestingly, this cell in the adural zone membranel, they separated by a gap, so composed of three frontal membranel and about 19 ventral membranels. This cell have about 4 micronucleus and 1 until 4 micronucleus that attach to the micronucleus. We also can observe one row of left marginal cirri and one row of right marginal cirri. Here we also provide the cyst from of the specimen. We could observe two kinds of cysts. First, the cysts that not possess of external and internal cyst layer, and the second one is the cyst that can possess external and internal layer cyst layer. So because of the present distinct gap between frontal and ventral adural zone membranel and also the presence of buccal lips, the absence of dorsal kinetic, caudal cirri, transverse cirri, as well as parallel membrane and the buccal cirrus, we assign our population to the genus Schmidingerothrix. Compared with other congeners, these species have two prominent frontal cirri, while in the extraordinaria they only have one. In the salinarum and salina they only have they have three frontal cirri, and then in the elongata they doesn't have a frontal cirri. And the other thing, our population have three frontoventral cirri rows, same with the salinarum and salina. And the other thing is contractile vacuole. Only the Schmidingerothrix extraordinaria that have contractile vacuole. For the cortical granules, our population doesn't have cortical granules, similar with the Schmidingerothrichidae. So based on the morphological, based of this morphological character, we can see that our candidate species share similar characters with Schmidingerothrichidae and salina, but also possess different character with them.
So, from the phylogenetic aspect, as you can see in this phylogenetic tree, that is our candidate of new species from Schmidingerothrix, they are clade together with the rest of congener from this genus. And our candidate of new species, they are clade together or close related with the Salinarum and Salina. And this topology of the 4 species from this genus is supported by the presented similarity and the nucleotide difference, which we can see that in the nucleotide difference and presented similarity that our candidate of new species closer to Schmidingerothrix salinarum and the Schmidingerothrix salina instead with the Schmidingerothrix elongata. And you can observe that in this tree, there is some topology or polytomy topology that happen within the Stichotrichida member and Schmidingerothidae member. The phylotomy occurs because of one is can be lack of sufficient data, in this case of the taxon numbers, and also the lack of the character that we use in this analysis, which is in this analysis we use the SSUR DNA only, that it can be that the SSUR DNA it's not sufficient, it's not effective to explain the real relations between the Stichotrichida member and especially within the 3 Schmidingerothrix member, Salinarum, Salina, and our candidate of new species. And then the next reason why polytomy occurs, it can be happen of multiple simultaneously divergent event that can leading to rapid speciation, especially in this three congeners, or sorry, in this 3 member of Schmidingerothrix genus, which is this end of our presentation. Thank you for your attention.